Cytokinins. History. In 1930, Gottlieb Haberland discovered an unknown compound present in the vascular tissues of plants which induces cell division. In 1940s, von Overbeek observed that milky endosperm from mature coconut is rich in compounds that promote cytokinesis. Miller et al. In 1955 isolated a substance in crystalline form from autoclaved herring sperm DNA and identified as 6-furfuryl aminopurine and named it as kinetin. The first natural cytokinin was discovered by Leatham et al. In 1963 and he named it as zeatin and it was isolated from unripe maize kernels. About 18 cytokinins have been discovered, for example, dihydrozeatin, benzyl adenine, etc. This is the 6-furfuryl aminopurine. Chemical nature of kinetin. Strong 1956 suggested that kinetin was an adenine bearing a furfuryl substituent at 6th position. Application of cytokinins. Cell division. The most important function of the cytokinin is to induce cell division. Cytokinin along with auxin induces cell division, resulting in the formation of callus in the culture medium. Number 2, cell enlargement. Cytokinin also causes the cell enlargement. The cell enlargement due to kinetins is partly due to more take-up of water and partly due to elasticity of the cell wall. Number 3, morphogenesis. Skoog and Miller, 1957, observed dramatic response of the cytokinin in the differentiation of organs from callus. In case of tobacco pith, if we add balanced quantity of IAA and kinetin, it results in the production of an amorphous and undifferentiated callus. And if at this stage, if we increase the concentration of cytokinin, you will see it promotes shoot bud differentiation, and by increasing the concentration of auxin will induce root formation in the callus. You can see the balanced auxin and cytokinin is resulting in the organ differentiation. Dormancy. Cytokinins have been found quite effective in breaking the dormancy of seeds and some other plant organs. For example, Lactuca sativa requires red light for breaking its seed dormancy, but its effects can be substituted by the application of kinetin. Counteraction of apical dominance. Cytokinins have been reported to induce lateral bud formation even when the apical bud is intact. Thus, cytokinin reverses the auxin-induced inhibition of lateral buds. You have studied the apical dominance, the practical application under the head auxin. Initiation of interfascicular cambium. Kinetin can induce formation of interfascicular cambium, that is, the cambium developing in between the two vascular bundles in the dicot stem. Richmond and Lang effect. It is also referred as delay in senescence. It was reported by Richmond and Lang in 1957 while working on the detached leaves of Xanthia. In this case, kinetin was able to postpone the senescence for a number of days, that is the degradation of chlorophyll and breakdown of proteins, which normally occur with the aging process of the leaves. You can see substitution of short day condition. If a short day plant treated with cytokinin and kept under long day condition, the flowering takes place. So here, cytokinin is substituting the short day conditions. Abscisic acid. F. Edicott in 1963 isolated from the young fruits of cotton an active compound as abscission 2 and was named as abscisic acid. Waring in 1963-64 is studying a compound causing the dormancy of woody plants, particularly that is Acer pseudoplatanus, and named it as dormin. Later on, both abscission 2 and dormin were put together under the head abscisic acid. This is the abscisic acid, a growth inhibitor. Translocation of abscisic acid. Analytical studies have revealed that abscisic acid, or ABA, move through phloem, xylem, and also in parenchyma cells. Role of abscisic acid, bud dormancy. Abscisic acid accumulation is responsible for the winter bud dormancy in the tuckweed plant. Senescence. Senescence of leaf promoted by ABA, that is the loss of chlorophyll, decreasing rate of photosynthesis, and changing the metabolism of nucleic acid and proteins. That is accelerating the senescence. Abscission. ABA accelerates leaf abscission in cotton plants. Flower initiation. In certain short-day plants such as Forbitis and Fragaria, ABA induces flowering during long days. Stomatal physiology. ABA alters the plasma membrane by affecting a change in a bioelectrical potential across them and an efflux of potassium ions. You can see the turgid guard cells, stomata are open. Potassium ions are taking in, and then the facet guard cells, the potassium ion are going out and resulting in the closing down of the stomata. Release of ethylene. It is now well established that ABA stimulates the release of ethylene. Counteract gibberellins, that is GA. ABA counteracts many effects of GA, such as induction of hydrolase in barley seedlings. You can see the difference. Ethylene. History. Observation of Neljubo, 1901, that ethylene gas can remarkably alter the tropic responses of fruits. Denny, 1924, reported that ethylene is highly effective in inducing fruit ripening. Ultimately, Gain in 1934 established that ethylene is actually a natural product for ripening of fruits. Ethylene releasing substance called ethyl, trade name, or ethepon, which is the common name, is commercially available. And you can see the various stages from green to ripened banana. And here, at the time of ripening, there is a production of ethylene in banana. Characteristics of ethylene. Ethylene is a natural gaseous plant hormone synthesized in ripening fruits, flower, leaves, and roots. Maximum rate of ethylene production occurs during the period of maximum respiration, that is called as respiratory climacteric. Role of ethylene. Ethylene inhibits elongation of stem, causes swelling of node, and nullifies geotropism. Ethylene retards flowering in most of the plants but accelerates in pineapple. Ethylene increases the number of female flowers but decreases the number of male flowers. Ethylene accelerates abscission of leaves and flowers. Ethylene is a ripening agent. It is formed in large quantities in ripening fruits. Few other growth hormones are morphectins, malic hydrazide, that is MH, then chloromycoate, LR85, amino 1618, phosphan D, coumarin, scopolamine, and para-ascorbic acid.